Okay, welcome back all. A very cheerful and breezy evening slash slothful afternoon, or say a warm sunshine morning to all of you. And this is the last session today, day 2, and we are almost halfway through the conference. And it's really exciting to see a 3-speaker lineup for this session. I'm Amir Khan from CCMB, and with me, Dr. G. Umapati, Senior Principal Scientist and Group Leader, CCMB, will be hosting this session. And I'll just brief you for today's session. We have Dr. Budhan, who will take us to opportunity and challenges in assisted reproductive technologies for endangered species. And then we have Dr. Ursula with us, who will be talking about contraception in wildlife animals. And then lastly, we have Dr. Douglas, who will be taking us to how to develop fertility control tools for wildlife management. So now I invite Dr. G. Umapati to introduce our first speaker of the session. Over to you.
Yeah, thank you, Amir. Today we have once again good morning, good afternoon, good evening participant. I welcome Dr. Budhan. Budhan Bukharendi completed his Bachelor of Veterinary Science in India in the 1987 from Chennai, and Masters and PhD in Animal Sciences at the University of Maryland in USA. He has been working at the Smithsonian Conservation Biology Institute since 1992. He leads the ungulate conservation program at the SCBI, where he manages research on several rare and endangered species. His research focuses on enhancing animal management, health, assisted reproductive technologies, stem cell biology, and genomics for assessment of genetic diversity, inbreeding and relatedness of individual animals in zoo, managed and private collections. This research team was responsible for first ever successful artificial insemination in Persian ongors, Peskovsky horse, and most recently scimitar pond oryx. He is the chair of the Association of Zoos and Aquarium Biobanking Scientific Advisory Group. He is also a member of IUCN Euclid Specialist Group, IUCN Topic Specialist Group, and IUCN Deal Specialist Group. Further, he is a member of several AZA taxon advisory groups, including equids, cervids, antelope, and tapirs. He also serves on the board of Conservation Center of Species Survival, a non-profit conservation organization. Budhan has published over 100 peer-reviewed manuscripts on diverse topics and monitored numerous post-doctoral fellows and students. I welcome Dr. Budhan for his talk on assistive technology in wildlife. Thank you for accepting our invitation first. Yeah, once again.
Thank you. Thank you for the introduction, Umapati, and thanks for the invitation too. Hello, everyone. It's always a pleasure to be part of these meetings, and especially when it's hosted by India. I always feel that a strong connection back to the country. So as part of this session, and I know in the last couple of days, the number of outstanding presentations have been made on various aspects of wildlife conservation and ongoing efforts in both in India and elsewhere, what people are trying to do to save a number of these species. So for my presentation, I want to focus on just an update. It's a very difficult, broad topic. It's a sort of a disclaimer, very likely that I'm going to miss somebody's important work. It's too much to cover, but I just want to give a snapshot of what is being done for different taxonomic groups. And I also see that as an opportunity how people either in the wildlife world or the zoo world, or even the universities can really facilitate the ongoing efforts to preserve some of these species. So as Umapati said, I am a staff scientist at the Smithsonian Conservation Biology Institute. Some of you attended Dr. Janine Brown's presentation yesterday, might have gotten a glimpse of what we are and where we are located. I'm going to skip some of that. So for the outline of my presentation itself, I'm going to sort of run down a few introductory topics, basically quickly go through what's the status of the challenges with wildlife in the natural world. What role do ex situ conservation or ex situ populations play with respect to species conservation, and some of the challenges that we are dealing with. So I see assisted reproduction as a tool to facilitate improved reproduction and animal production in an ex situ managed program, because we all dream about a day when we want to be part of a reintroduction program, taking an animal that is either endangered or threatened with extinction, bring them in a managed situation, grow the population, and release them in the wild. So that's a dream for everyone, and not a number of people get that opportunity to be part of the dream, but you can contribute to it at some point. And so I think that I hope this presentation will encourage you to consider some of those. I do want to talk about a few topics that are sort of novel approaches. I don't know if this truly has a immediate application to endangered species, but again, in the world of science, we need to stay relevant in the context of technologies and technology development so that when it's ready to be applied, we're able to use it. So this is all known. This is an ongoing threat day to day. The most recent climate change talks, you see that the biggest challenge is loss of habitat and habitat destruction. I think the blame falls squarely on us as people, because we are the ones who are making those decisions. And for our own comfort, we are also destroying habitat for the species. Poaching, illegal poaching and hunting still continues despite a number of countries investing several billions of dollars to prevent it and all the laws in place. And when it comes to oceans, I think this is another massive area of study and warranting much more attention because of the acidification and loss of marine habitats. But it's not all gloom and doom, right? With respect to bringing animals into a managed situation, there are extremely, extremely exciting stories where animals have been reintroduced. California condor is one, the Kowalski horse that's been released and reintroduced, the Wyoming toad, the black-footed ferret, and also some of the coral restoration projects that are ongoing, which again is as good as the reintroduction of species back into the natural world. So it is exciting, it's possible, as long as we are committed to pursuing that. When it comes to ex situ management of zoos, there are a lot of these programs. Several of you are familiar with the associations that control or regulate organizations. We have, for example, in India, you have the Central Zoo Authority, which is managing a lot of the zoos and the operations, and the Forestry Department, which is also managing the wildlife populations. So these are some of the programs that we are oftentimes engaged in with the AZA, Association of Zoo and Aquariums, SSP or the SAFE programs, the European counterpart, EEP, and you have the CIESA and all other. So this is a program, something similar to the Species Survival Plan, is a group of volunteers who manage certain species with the idea of maintaining sustainable populations over a 100-year time frame. And that is purely based not on numbers, it is based on the genetics and the genetic quality of those animals that we are producing. One of the simple goals of these ex situ programs, it's an insurance collection, right? It's like you getting an insurance policy, a life insurance policy for the future if something were to happen to you or to your animals. As I mentioned, the key thing is genetic management. It's not the numbers game, it is the genetic quality of the animals. I think when you bring animals into an ex situ collection in a human care situation, it is very important that those animals also become part of a research population. Because I think it's a travesty if we don't use these animals to learn more about them, because that knowledge is so critical when it comes to recovering a species and helping them with reproductive problems or even health issues. We all talk about genome resource banking. Again, the sky is the limit for it these days. Again, we don't know where this technology is going to take us, but systematically banking tissues, gametes and somatic cells, all of those can be of value. Finally, the outcome is really to lead up to reintroduction itself. But unfortunately, most of the species that we have in ex situ collections are not self-sustaining, meaning that they're not reproducing. We don't have adequate number of founder animals to maintain a genetic stock for a long period of time. And what we have been doing is to explore some new options. And this is an organization that we are members of, the Conservation Centers for Species Survival, where we are exploring partnerships with private sector. As some of you may know, there are a lot of private ranchers in Texas. There are thousands of these endangered species that are managed there, either for conservation purposes, and some of them are engaged in some hunting business operations too. So we need to explore some new opportunities because space in zoos is always going to be limiting. This is our center. We have about 30 to 100 acres of land, but still we know at one point we are going to explode in terms of the space requirements. So we need to look at all other options. So when it comes to ex situ management, we absolutely want these animals to breed naturally. I think that's the best for the species, for the collections. And that's also the most efficient because we know any assisted reproductive technology that exists now does not give you high levels of return. I would say at best we are at about 30 to 40% or 50% in some cases. But most of them hover around 10 to 20%. Versus a natural breeding, if all things are facilitated and optimized in terms of husbandry and health, you're guaranteed 100% in most of the species. So it's really very difficult to beat natural reproduction. So these are some of the common techniques most often considered, discussed, and known and studied. So this graph, basically the vector graph shows that there are a lot of the species. Again, obviously I cannot show everything here, but quickly looking through, the green circles basically suggest that these are some of the taxonomic groups that most of the work is being done, either because of the interest. When you think about mammals, most people recognize more mammals, but when you think of lesser species, the public don't recognize it. And sometimes even the access to some of those other lesser species is very challenging. Most recently, there's been a lot of work on sharks and rays. So it's really interesting to see that is evolving. And very limited studies in some of these model systems, if you were to look at. I wouldn't say some of this work has been done on endangered species itself. I mean, there is some model species that could help us with respect to endangered species. This work is all done, right? I would think that most of the folks who are attending the session know that for a large number of mammals, techniques for collecting semen have been standardized, and there are still some that we need to work on. Again, most of these technologies require anesthetizing these animals, which are high risk, but as long as people are comfortable, people are well trained and prepared, taking all the safety precautions, this is a safe procedure to do and a fairly successful procedure to collect considerable amount of sperm to work with, a semen to work with. More recently, there have been techniques to collect semen by catheterization. So instead of using electrostimulation, especially the technique like this would be very applicable for field situations where you don't have to take a whole lot of equipment. Once you anesthetize, you can pass these catheters and get relatively good concentrations of sperm and good quality semen too from a number of species. And the same approach has been expanded beyond cats, felids, to other larger species. And the output is still comparable to electrostimulation or electroejaculation. Good number of sperm, good quality sperm, comparable quality. And this again, where people are hesitant to induce or use electrostimulation, this is a pretty safe procedure. Again, sterility is such an important factor to consider. You don't want to introduce infection into the reproductive tract. So again, training is key, and following certain protocols is extremely, extremely important. And I feel that in the years to come, this approach is going to be developed for a lot more species just to make it more convenient. When you look at our assisted reproduction, I would say the felids have benefited the most for obvious reasons, because cats are very charismatic and people are very willing to support research and invest funding in these areas. So the next couple of slides are basically data slides. Sorry, it's very dense. I just basically took from some of the recent reviews rather than go through the specifics of the detail and the procedural details, but I'm happy to answer those questions towards the end. So here's a review that came out in 2020. You can see that when you look at the success rates with respect to small body cats, medium or large body cats, you find that the rates of success in terms of pregnancy is highly variable. And I think part of the problem is also the number of animals that you work with. This is an ongoing challenge for most conservation biologists. most conservation biologists, because we don't get access to adequate numbers to do systematic research in a way that it is done oftentimes with livestock, where you may have 30, 40 animals in a study. So the success rate is very challenging to really nail down because there are so many factors that contribute to the success or failure of these efforts. But going down, you see that most of the efforts are being focused on laparoscopic intrauterine insemination. In the last several years, people like Bill Swanson at Cincinnati Zoo have also developed the laparoscopic oviductal insemination techniques, which have also resulted in some relatively good success where live offspring have been produced. The advantage of the oviductal insemination technique is the amount of sperm that you're going to need. If you're using, if you're applying intrauterine insemination, you're going to be using several million sperm for a single insemination, versus in the oviductal transfer, you're taking the sperm so close to the egg, where in the oviduct, you can get away with a couple of million sperm. So extending the lifespan of the samples that you have, either fresh or frozen samples, over at multiple doses is such a key component. So again, you can see the success rate itself is highly variable, right? It's 0 to 100. So we just have to be mindful of it. It's not a highly reproducible option, but this is still one of the best options that exist for females. This is a more recent one that in the Jaguar group from Cincinnati with the university in Brazil reported. Once again, it's an oviductal AI technique that was used. The first one that was produced, unfortunately, this cub didn't survive for long, but at least it demonstrates that the technique works, and more with repeated work, additional work, they should be able to get more offspring on the ground. When it comes to embryo transfer, a number of species in felids have been successful. In small cats, a lot more successful than the larger cats. Most recently, our own institution is engaged in successful embryo transfer in cheetahs. Again, the approach is it's laparoscopic, where you do all of the oocyte aspiration, and then you do in vitro fertilization. And then once again, we use a different set of donors in most cases, or recipients, or we may go back to the donor itself, where once again, we do a laparoscopic procedure to do a oviductal transfer of those embryos to come up with result in a pregnancy and live offspring. Again, we have a lot more examples of 1 or 2 successes, and I think the challenge that still remains in the world of cats, cat reproduction and assisted reproduction, is repeating those results, making it more consistent, more reproducible, and more translatable, that we can apply these approaches for a number of species in cats and consistently get pregnancies, whether it is artificial insemination or embryo transfer. So these successes should not stop further research on these animals. I think it's very key to continue, push forward, and get as many animals and institutions to participate in this research, and getting the authorities, convincing the authorities to support these research itself, because as I mentioned before, every animal that's in a managed situation in human care, we see them as a research resource, and they should be utilized as long as it is done safely, and there are enough regulatory systems in place to ensure that. When it comes to the idea of cloning, because this comes up all the time, it's very tempting. It's a very sexy procedure, but personally, I'm not a big fan of it, simply because purely from a conservation perspective, ex situ management perspective, genetic management perspective, I see artificial insemination, embryo transfer might be more applicable than cloning, especially when you look at the success rates that we see. Getting into a blastocyst stage in a dish is one thing, but getting live offspring is a completely different ballgame. Unless we see more of this happen, more reproducibly, and not have to transfer hundreds of embryos to get a single offspring, I think, in my opinion, I would say cloning is still something that is to be developed, but I don't see that as an immediate application, a tool for immediate application. For artificial insemination antelope, I'm just going to use an example. Oftentimes we come up with the question of how do you work with these animals without using anesthesia, because most of the previous techniques that have been reported have used anesthesia, which is general anesthesia, and do very similar procedures like either laparoscopic insemination or transrectal insemination like we do in cattle and in horses. So we have been working on minimizing the dependence on anesthesia. I'm going to show you a video. I hope it works well. This is the recent work that we did. This is one of our barns. We have these chute systems that we have built. So these animals are conditioned to go through these chute systems on a daily basis. Routine animal management. So any given day when they walk through the chute system, they are going from one end of the barn to a pasture for a food access. So on a day that we want to work with, we are able to bring them into the same situation, but we close the doors. You can see the doors that's closed here. And now we are able to push the animals back into the chute system so that we can get to work with them. Again, it's a lot of training, conditioning, desensitizing that needs to go through. And again, having a lot of the safety checks, systems and backups. Here we use a hydraulic device. For the animals in here, we are able to restrict the animal movement. And we, oftentimes in a larger animal, we even lift the animal off the ground a few inches. Here we're just marking the horns so we can visualize and see who those animals are from a distance, and to collect fecal samples. Janine Brown and several members of this audience already are familiar with this technique, where we're able to assay the hormones. Most of our work, if you're thinking about artificial insemination, we do some of the hormone assays after the fact. We just come back after we do the AI, we come back and look at the hormones to see where those females were in the reproductive cycle. So that sort of gives us an idea what this outcome may be, whether we are going to get a pregnancy or not. So samples, we use our staff, animal managers, animal keepers who help us with all these projects. Over 350 samples that were collected were analyzed. Obviously, my colleague, Janine Brown, and her team helped us with the analysis. And we used frozen sperm. We had the semen that was collected and banked about 15 years ago. Again, the value of genetic banking. This male is no longer alive, but to be able to bring those genetics back into the population. So there is no anesthesia involved in it for the AI procedure itself. We once again catch the animals in the restraint, the hydraulic restraint. We had 10 animals in the trial that we use for the artificial insemination itself. And it's a very simple technique. We catch these animals, pass a catheter very close to the cervix. Rarely we are able to go through the cervix in this situation because it's pretty blind. We deposit the semen and we let go of the animal. So it takes about a minute or two to get this done. And we were fortunate enough to have at least 3 pregnancies to start with out of the 10, and 5 animals responded to the hormone protocol that we had set out to do. So 5 out of the 10 responded to the hormone protocol. 3 out of the 5 that responded actually showed early pregnancy using pregnancy-specific protein B assay, but then 2 females then eventually gave birth to live offspring. So the idea here is no anesthesia involved in it, integrating artificial insemination techniques into ongoing animal management procedures. So the key is infrastructure, having the right people, having the right training, and conditioning these animals to help you with these procedures. Where do we see with this procedure, where are we going to go with this procedure? As some of you are aware, this is a species that was recently reintroduced in Chad. Animals are being organized and assembled in the UAE and also handled constantly in Chad before the release. We feel that we may be able to take frozen sperm from a number of animals from Europe and in the US and artificially inseminate females rather than transport a lot of male animals, which is expensive and risky, and get some more new genetics into the population, in the wild population. So that's really our strategy. We'll see in the next few years how this becomes a reality. And with respect to cervix, embryo transfer, artificial insemination has always been shown successful. And this is a recent report that was published by our colleagues at the Zoological Park Organization in Thailand and my colleague here, Pierre Camizzoli, where they did embryo transfer with the Brava antler deer that resulted in live offspring. Again, in vitro fertilization are done, oocyte aspiration laparoscopic. After fertilization, embryos are back again through oviductal, laparoscopic. I mean, this is a laparotomy because that's one easy way of visualizing, but it's oviductal transfer resulting in birth. When it comes to rhinos, again, the last few years, I think you also listened to some of the speakers in the session about work that's being done on rhinos. It's interesting because I think even though there's a lot of interest in rhinos for a long time, there's not been a significant infusion of funding until the northern white rhino crisis was revealed. And now we have donors who are willing to invest millions of dollars into this research. So irrespective of the outcome for the northern white rhino, my stand on this is the amount of money that has come in to study rhinos in general is going to allow us to do extensive amount of assisted reproduction for the species because of the biology that we learned for all of the species. So it's a very interesting model, but a lot of work is being done. So I'm looking forward to see what comes out of all these efforts. A number of groups that are doing outstanding work globally. So when it comes to semen collection, the rhino anatomy, as most of you are aware, the testis is the intra-abdominal. But again, with semen collection, we do need specialized tools to do it. Electrostimulation is still a good technique for semen collection with rhinos. And in my earlier slide, I talked about urethral catheterization. That is also a good option for rhinos. This is a review. Again, look up the review article that was published by Pennington in 2019 for more details. Most of the animals, you're able to get excellent quality samples, ejaculates, spermic ejaculates from them. And even after postmortem, so when you lose an animal, please make sure that you get access to those tissues, reproductive tissues, because that genetic material is still extremely useful for future population management and assisted reproduction, because sperm and eggs and ovarian tissue and testicular tissue are all still viable and could be used for various other technologies. In terms of ART itself, you can see it's a summary, a nice summary of all the different things that are currently being done or has been tested or being improved. So you have anywhere from artificial insemination, semen collection, ovum pickup, ICSI, IVM, IVF, all these things are being implemented because anything you have as a tool to help the species, I think we should apply. And this should be a strategy for any species. It shouldn't be restricted to rhinos. Any species that you have access to. If something were to happen to that animal for medical reasons, please work with your colleagues in the research world and our universities close by to see what you can do with respect to utilizing those materials. Some successes that came out recently through artificial insemination in the southern white rhino. So some of this basic biology hopefully will lead to the recovery of the northern white rhino itself. This is another cool area. Again, just to get some folks in the audience excited about, you don't have to, all research need not focus on mammals. There are so many other species. I listened to a presentation early this morning where one of your own colleagues was working on reptiles and turtles. So plenty to do. So again, why care about sharks and rays? A good number of species are considered not critically endangered or endangered. So this is again another group that needs attention and research. With respect to aquarium function, I think this is where the aquariums can come into play, and not some zoos may also have small aquariums that could also contribute. A number of species have been studied, and I think this will be a field that is going to explode in the next few years, given what is happening to our marine ecosystem. In terms of semen collection, semen can be collected by abdominal massage technique. We can also do some cannulation and aspirate sperm. And obviously, when you have animals that die, or even when you find them in markets, if it's relatively fresh, I think that's a good source for collecting gametes from these species. Collections can also be done in the field. This is in the wild, a sand tiger shark assessment, ultrasound being done, and also semen collection done out in the field. And this is the work that Cesar, my colleague, dear colleague, Linda Penfold and her group is developing. Sperm from the shark. It's a really cool morphology. Again, anybody who's studying reproduction, all these morphotypes excite you when you go from one species to another. But then the better part from a reproduction perspective is trying to see, once you collect semen samples, what can you do with it? So here's a small spotted shark data set where you can maintain chill samples for 36 days. That's impressive. Very few species you can do that. And again, depending on what conditions you're using that for. Here, another data set for the white spotted bamboo shark. You can see that, again, the samples can be cold stored for extended period of time. If you're considering artificial insemination, you can just simply use chill semen. Fertility assessment, using chill semen here. What they found is that the semen can be used for a long period of time, and they will continue, the females will continue to fertilize their eggs and produce a number of egg sacs. So this is, again, a very fascinating finding, which could have broad implications for sharks. With respect to cryo preservation itself, you can see a lot of different cryoprotectants or diluents are being used. On average, you're getting about 40% of what you're starting. So in my assessment, 40 to 50% is a very good start for a new species. Obviously, researchers can further improve the quality, postdoc quality. Frogs, same issue. There was another presentation this morning talking about amphibians and the amphibian decline that's ongoing globally. This is research that is being done at Smithsonian in Panama. We have a conservation breeding program set up there to rescue these frogs from the wild and bring them into a captive situation. And here the data shows that some of the options, techniques that we are developing for collection of sperm using hormonal stimulation. You can see this particular treatment with the 4 micrograms of GnRH, you have a fairly high concentration of release and a sustained release of spermic volume, urine, that we are able to work with. And that's comparable almost to the Ampiplex scenario, which is a natural way of stimulating ejaculation. This is simply to show the quality of samples that we are able to get in these systems. And so I think we do have a fairly extensive bank collected frozen sample in Panama as part of this program. There's also artificial fertilization work that's being done. Here's an example of the work that is led by Andy Kuba, and they're working with the critically endangered wild toad. They're able to collect spermic urine, get the eggs, and then artificially fertilize them and produce tadpoles. Again, this is a success story for amphibian where animals have been produced in the lab and then have been released in the wild. So it's really fascinating and it's promising to see all this. Coral is another area. As I mentioned, most effort that most people are aware of what's happening in terrestrial landscapes. The marine landscape is being destroyed every minute, and either through overfishing or some of the changes that are happening in the marine water systems itself. And my colleague, Mary Hagedorn, and several of her collaborators have been focusing on coral conservation. And I'm just thrilled to see the amount of work that they've accomplished in the last few years and how fast this field is growing. As we know, a big challenge is the coral bleaching that is happening. And coral reefs also maintain a huge diversity of marine species. So more reefs destroyed, more marine species that are destroyed automatically. So semen collection wise, sperm is basically, you basically try to collect these samples during typical spawning cycles. And here's a spawn event, and you're able to go in, either if you have in a lab model, you can do this work in the lab, or somebody have to dive deep to get to these spawning events. So techniques have been developed to freeze these coral sperm. And here's an example slide from a published data set where coral larval settlements you're looking at after you fertilize them using fresh or frozen sperm. You have the larvae forming, and then you look for the settlement of those larvae in a dish. And you see that it does lead to comparable fertilization and larval production. And here's a larvae that new coral that has been formed using frozen sperm. and to produce a larvae and settlement. And this is, we basically call it as assisted gene flow, so that you can move some of these materials, sperm and larvae, from one area in the marine ecosystem to another to repopulate those ecosystems. As I mentioned, in the last just 10 years or so, the number of species that have been collected in bank, this is a global effort. Hagedorn and group have done a tremendous amount of work in training people, getting them excited about all of these to have regional banks for coral species. So it's really cool to see this. Anyone interested in this field, please reach out to Mary Hagedorn. You should be able to locate her email very easily. So what other technologies have been developed? This group has developed methodologies for freezing coral larvae, coral embryos, and again, size is a challenge, and people who are working with embryos, survival of frozen thawed embryos is really dependent on how fast you are able to thaw that material. So they've been using laser systems to thaw these, and also they're working on a high throughput laser system to thaw several hundreds of these embryos in a short period of time. And in coral, it's not just the coral, the sperm and the eggs that matters. The symbionts are the key things. The colors that you see, the organisms that inhabit the coral and keep them alive are the symbionts. So even if you're able to freeze the sperm and eggs and embryos and the larvae, to maintain a healthy coral system, coral reef, you also need the symbionts. And the same group has also developed methodologies to cryopreserve symbionts so that you can repopulate them. So some of the work has been summarized in this book chapter, and that came out a couple of years ago. So please look that up if you need more information. What are the other technologies? These things have been around for a while. Desiccation, because how the challenges people have with respect to preserving gametes and embryos in liquid nitrogen. Maintaining a liquid nitrogen collection and repository over the long period of time is expensive, time consuming. You need people, you need backup systems, alarm systems, everything. So a number of labs, this is again model systems, right? The mouse model, where most of the work seems to happen initially. Desiccated sperm, keep it in ampules. You don't need liquid nitrogen storage, but you also need advanced techniques like sperm injection to produce an offspring. Here's an example where desiccated sperm was mailed to a different lab, literally mailed in the post. And then those sperm were reanimated. ICSI produced live pups. So it's not science fiction. It can be done, provided you have the rest of the biology and technologies worked out. And it needs to be transportable in some cases when it comes to wildlife applications. So we have to kind of keep that in mind. Yes, these are techniques that we need to develop, assuming that in the next 5 to 10 years, a lot of these added techniques for producing live embryos and live offspring will evolve over time. Here's an example of rhesus macaque, the same approach, desiccated sperm is used for ICSI and producing blastocysts. What are the other options? Dry preservation, tissues. We have in our lab, Pierre Comizzoli's group has been working on desiccating ovarian tissues. Here you can see desiccated, reanimated, and the tissue is maintaining relatively good morphology. And likewise, testicular tissues, you can go through the same process, desiccation, reanimation, and you find the tissues are maintaining fairly decent histological structure and viability too. In terms of gamete rescue, this is again coming back to the idea of when animals die for whatever reason, health, some accident happened and the animal dies. I think these are techniques that are to be developed, and we are using model systems like cats and dogs and maybe sheep and goat. The idea behind here is when you get these animals, it's very rare that you get a lot of oocytes, mature oocytes from these ovaries. But every ovary has thousands and thousands of premature follicles, developing follicles. So if you're able to harvest those early stage follicles and come up with ways to grow them in vitro, mature them, they potentially can lead to the production of embryos down the road. So it's a very valuable, powerful tool for rescuing gametes. And This is again being developed in our lab for cats and dogs, for canids and felids, endangered felids. So let's see what's being done. Some of the options we have is to grow them on Agarose Blocks. This is a technique that was published several years ago in testicular tissue in mouse. And you can see that these pieces of tissues that are being grown and these green spots are the viable follicles. When you grow them in cell inserts where these tissues are immersed in the liquid, the media, they don't do well. But if you grow these tissues in a liquid gas interface, they tend to do a lot better in terms of the number of viable follicles itself. You also see that the proportion, the number of follicles that are surviving over a period of time, long-term culture, you can see over day 0, day 3, and day 15. By day 15, most of them are disintegrating or degenerating. So this is in the dog model. Interestingly, in the cat model, similar culture system maintains much better integrity. So again, focusing on the species differences, species-specific differences is also very important when you're developing tools. What works for the cat doesn't work for the dog. We're also growing follicles, isolated follicles in an alginate system like encapsulation. You find that we're able to grow them. You can see in the dog oocyte, starts with the dark cytoplasm over time, it becomes pale and it dies off, the follicle dies. And we suspected that it could be a problem with the communication, the granulosa cells, the cumulus cells in the oocyte itself, and leading up to the antral collapse. And how can we improve this? Some of the studies that's been done in our lab, we looked at adding activin in the presence of FSH. Certain concentrations of activin does help with the antrum formation. So we know we can push these systems a little bit. And here you can see in this picture that the communication, the junctions are more intact than here where it is very fragmented. Where we're going with it, rather than having static cultures, we are exploring microfluidics. So you are trying to mimic what is happening biologically with a slow movement of media over the tissues. Here you can see with different flow rates, 10 microliters per minute to 2 microliters per minute flow rates, you can see that they're not much of a difference, but you see some patterns are emerging. And when you look at the primordial follicle diameter, when you culture these tissues with a 2 microliter per minute flow rate, there's a slight difference, statistical, but a slight difference in primordial follicle diameter, suggesting that some of those follicles are surviving better. So in summary, I threw a lot of things at you. Again, this is a very broad topic, but I hope I just got you thinking about it, and there's plenty of review articles that are available for you to look at. So I would say development of assisted reproductive techniques is warranted in a number of species. We have really literally scratched the surface itself. And to maximize to the use of ex situ collections, as I say, I think any institution or any country that has animals in human care in a zoo situation or in a breeding program needs to make those animals available for systematic research to help the species. And I think it can be done. There are a number of systems in our own institution. We've been able to do it for the last 20, 30 years now. In Europe, it's happening. I know in India, it's happening too, but still a lot could be done. The main reason I'm saying that is the way international zoos are going, it's very likely that a number of species that are endemic to tropical countries are no longer going to be part of the zoo collection because of the difficulty in obtaining new genetic stock and some of the disease issues that we have to think about in importing animals. So I think it's an imperative, it's absolute necessity for countries in tropical areas where most wildlife is threatened to really increase their activity in research and propagation of these animals. If you have critically endangered species or endangered species, to come up with these plans to maintain a healthy population. It's easy to move those animals in your own country. You can get more genetic stock if you need to, and you can maximize the quality and the quantity of research that can be done. I think that sperm cryopreservation and artificial insemination remains one of the most popular ARTs for endangered species. And other techniques may evolve and may be applied further later on. And as I mentioned towards the end, with respect to the in vitro techniques, I think these are emerging technologies that we need to pay attention to. We need to invest time and energy and resources to develop. keeping in mind that those techniques may take maybe another decade or so to be applicable for conservation of endangered species. The work that I presented here is really a summary of a number of groups globally where people were willing to share, and most of the data is also published data. So please look up those information for details. I did not go into all of the details of what you need to do technically, but this is just to give you, it's a teaser, look at it that way. Thank you for your attention. I really appreciate this invitation and your willingness to spend the hour with us late this evening. I will stop sharing and take questions.
Thanks, Dr. Budhan. That was a nice presentation. And it's nice to see that shark, rays, and especially coral made it to your talk. We don't see people talking about, these species. So it's really great to see that. And yes, I mean, it's important that we should, utilize, I would say, the storage of animals in zoos to make more knowledge, to dwell into, knowledge and use ART and maybe share experiences of failure, especially. So we should learn, build on that. So such conferences made it possible. So really thanks for that. And yes, we do have, I think, 3 or 4 questions. So I'll read out for you. And if any panelist feels like they need to speak, so please unmute yourself and do it. So yeah, so first it goes from Dr. Narayan. So he's asking that he's very curious that is any work in insect, special one like bees, spider, especially on the ecology function for human advantage, example like age and other things. Then he said, thanks for the amazing presentation. A second question also here that it was effort to preserve tissue from various island nation with very rare species, example crested iguana. So I mean, yeah. So some complimentary question. Yeah.
In terms of the insect itself, again, as I mentioned, there's a lot of topics I couldn't cover. There's been some really good work that came out of a group in the US Department of Agriculture where they attempted to do a lot of freezing of bees in particular. And I think there are some groups that are working on spiders too. So that please look up the literature. I'm not 100% familiar. I don't have the information handy, but there are several groups that are working on these. It's not just for preservation of them. I think another fascinating point of this effort, the comparative aspect, Janine Brown talked about it too yesterday, and some of you at CCMB and other institutions are already doing, is the comparative aspect. Because there are a lot of these mechanisms that exist in other model systems in terms of preserving the viability of gametes, even in a live animal. Classic example I can think of is how bats maintain viable sperm for weeks. Simply by changing the osmolality of the seminal fluid. So there are a lot of benefits to come out of it. So please, if you're interested in other model systems, look for it. With respect to tissue freezing, I think the challenge remains when we talk about tissue freezing, most people jump to the conclusion that it's going to lead to somatic cell cloning, nuclear cell cloning, right? That seems to be the connection right now. Yes, it does lead to it, but at the same time, if it is reproductive tissues, you can also, as some of my slides towards the end are presented, you may be able to recover sperm from some of these materials. You can preserve testicular tissue or ovarian tissue. Yes, any methodology you can come up with to preserve tissues, either for reproductive function or for somatic technologies, please, I think we should try. If the science doesn't exist, look at what's been done in other species. Start somewhere and make your own. I think that's how it is. Yeah.
Thanks. Next question, Pratibha Verma. So she wants to know that while sperm collection, how to get rid of contaminated elements if they are present there, especially in rhino sampling. So yeah.
Oh, excellent. I think this is a challenge that we deal with, especially large animals. I see Safi's on the call here, who has a lot of experience both in Matthias, a number of people, you all have done this in the field conditions, right? It's not sterile. We know it is not sterile. We take all precautions to minimize the contamination. basically cleaning that space, flushing that refuse, and almost quote unquote trying to sterilize that area before you start the collection. In spite of it, you're going to see some contamination of that material. So some of the options that exist may be using some differential centrifugation, density gradients that are available. You may be able to eliminate some of the other contaminants that are in the sample itself. Beyond that, I would say, fortunately, animals have a fairly decent immune system that, despite what you put into them, depending on where you deposit the semen, if it's intrauterine, I would be very cautious not to put contaminated samples. If it is vaginal insemination or just close to cervix insemination, I think there's enough immune mechanisms that it gets rid of a lot of the contaminants. But again, I think the start, making sure that you start clean, gives you a much better result. But eliminating, bringing it to zero is very, very hard.
Thanks. So next question was from Bunte. So in embryo transfer in cervix, is percentage depicting IVF success rate upon oviductal ET? If it's
yeah, it's not great. Because again, it's a small number of animals that have been worked on. Even for the success that they had, again, this is one of those things that I mentioned and emphasized in my presentation. Single time success, or maybe 2 or 3 times success, is exciting. We all get very happy about it, delighted to hear that news. But our job is to continue doing that to make it more reproducible. It's at that point that technology becomes a tool, because you want to take a tool and help you do something. If it is still a trial and error system where you have 5 or 10% success rate, it's very difficult to say, hey, here's a reliable way to go about doing it, and you're going to get an offspring. So it's relatively small. I think it's less than 10 or 15% overall with respect to embryo transfer, even with the L-steer, because a number of procedures were done for several years in Thailand that resulted in 1 or 2 pregnancies there.
Great. So next question is from Dr. Jivamapati. He wants to know, have you able to cryopreserve organs? Any progress in this direction?
Cryopreserve organs?
Yep.
Oh, wow. No, I mean, there have been some discussions within our own group whether we should be looking at freezing testes as a whole organ system or ovaries as an organ system. I know there are some really exciting studies that are ongoing in the mouse model and even in the human model, where there are efforts ongoing here to figure out what best way to do it. But I'm not sure if anybody is trying in the wildlife world. I may defer that question to Sefi if he's aware of his side.
There's another question from Pratibha. So it's really long. So I would say if you can please unmute and then speak yourself, that would be, I think, great. Pratibha?
Pratibha is not.
Okay, cool. I'll just read out. It's very long,
Yeah.
but yeah, just bear with me. So viability after preservation and quality, like DNA degradation or increasing apoptosis. How long they can be stored without loss in quality and molecular level? In testes, IHS, no sperm were there in human. So at what age these testes should be dissected and preserved? If I understand right, that testes were dissected and preserved, but not directly cryopreserved. So that was.
Oh, okay. In terms of preserving sperm or gametes, even embryos, let's say, even cells, somatic cells, I think if the technique that you are applying to preserve that material is the right one, optimized for that particular species, and if it is stored the way it is supposed to be stored, the long-term damage to it in terms of DNA degradation and apoptosis is very, very small. As what we are aware of is, I think literature is out there too, is one of the biggest challenges, environmental radiation is a key thing, which it's very difficult. We are being bombarded at this moment as we are sitting around, we are being bombarded by environmental radiation. That's something that will continue to happen, but it's a very small proportion, very, very small fraction that is going to cause damage to the samples. The key thing is making sure you have the right technique for collecting and freezing the material and storing. Once you put it in a liquid nitrogen, you are able to hear me?
Yes.
Okay. Okay. Once you store it in liquid nitrogen, as long as you're monitoring those tanks with the temperatures, I think those samples are good for a long time. I'm coming back to the testers. Depending on what animals you're working with, there are techniques that are being developed for freezing testicular tissue, both from immature as well as mature animals. Immature, keep in mind, all you're going to do is early stage spermatogonial cells that you're going to be preserving. So you need to come up with technologies either by doing transplantation or grafting, xenografting, that you may be able to produce sperm, or even try to do some in vitro maturation and spermatogenesis. Technique that is still not refined beyond mouse model, something that needs to be done in endangered species. When it comes to mature animals, you have mature sperm, and there have been a number of publications that have come out where you're able to freeze the testicular tissue, revive that, rewarm or thaw that material, and you're able to get sperm. Obviously, motility is still going to be compromised, but if you're using technique like ICSI, you will be able to use that sperm immediately for producing an embryo, so that options exist. I think I covered all questions. I don't know if
yeah, thanks a lot. And thanks for keeping in time the talk.
Well, thank you, everyone. Really appreciate the opportunity. Thanks so much, Pathy.
Thank you, Dr. Budan Bukharendi. And we have the second talk lineup is Dr. Ursula Birchwood from Spivak Wildlife Services from USA. And my colleague Amir Khan would introduce her to the audience.
Thanks, Dr. Omapathy. Dr. Ursula earned a BS from Utah State University and DVM from Washington State University. After working as a veterinarian for several years, she earned a PhD from Oregon State University. Her research focuses on reproductive physiology of wildlife and population management tools, as well as nutritional and pharmacokinetic studies. She has worked in academic administration for over 23 years and currently serves as Director of Graduate Programs in the School of Arts and Sciences at the University of Pennsylvania. She is also the research advisor for the Bureau of Land Management, Wild Horse and Burrow Advisory Board. So I welcome Dr. Ursula, and I just learned that it's quite early morning there. So a warm welcome. I think you must lead this and then go ahead. Thanks a lot.
Thank you. Let me see if I can share my screen. Everybody see that?
Yes.
Yes.
Yeah, go ahead.
Well, thank you for the invitation to speak at this conference. And indeed, it's very early for me, but I understand it's very late after most of the conference all day in India, for those of you there. But I've had my shot of caffeine, so I hope that what I have to share will be of interest and stimulate some new ideas in the field of wildlife population fertility control, which is basically the opposite of what the previous speaker was talking about. After a brief overview of some of the challenges involved in managing populations of free-ranging wildlife, I'll focus on immunocontraception based on the antigen PZP, or porcine zona pellucida, specifically Spayvac. I'll share some results from trials with gray seals, a variety of deer species, African elephants, and horses. I'd also like to share what we learned about some immune responses from different horse trials and end with some ideas for future research. But I'd like to start by acknowledging that the key problem is really too many people, which reduces available habitat for native plant and animal species. Subsequently, some species may become endangered or go extinct, while others become locally overabundant. People here in the US are always surprised when I talk about contracepting elephants because they know that they're endangered, but they may not know about human-elephant conflict or fenced reserves. Local overabundance can negatively affect the environment and other species, and in an effort to preserve biodiversity, we resort to population management schemes, which are really Band-Aid solutions that don't address the key problem, but it's important to do what we can. Some examples of locally overabundant species include deer in North America. Too many deer can negatively impact forests, damage agricultural crops and private gardens, contribute to vehicle collisions, and spread tick-borne diseases. However, deer can also serve as important herbivores and prey in ecosystems. And for example, their fecal nutrient input increases growth of adult trees. The goal is keeping population sizes in check, and hunting isn't usually an option in suburban areas. In the US, free-ranging horses have few predators, which contributes to annual population growth rates of 15 to 20%. Overabundant horse populations can negatively impact the environment as well as other species, sometimes through direct competition, for example, with pronghorn antelope for water. Conflict between animal rights groups and cattle ranchers over limited resources on rangelands are also common. Primates typically have diverse diets, which help them thrive in a variety of environments. An introduced population of macaques in Florida, here in the US, grows at an annual rate of 11%. They've been known to steal items from people, which they can use to barter for food, and they can be quite aggressive. Zoonotic diseases are a concern. The list of overabundant populations is long. There are ways to control wildlife populations, but not without challenges. Hunting may be a popular sport, but sharpshooting in neighborhoods is not usually viewed as safe or acceptable. Physical terrain can make it challenging to gain access to animals, and costs of management differ depending on the treatment and number of applications required. For example, some vaccines that require boosters. Population reductions may be offset by increased survival of offspring if there are more resources available, or immigration from nearby areas. And the policies and regulations in different states and counties are further considerations. Regardless of the fertility control option chosen, there is not a one-size-fits-all solution. A variety of factors should be considered when designing a population management program, and I'll briefly go over each of these listed here. Considerations for site suitability when, for example, wanting to control deer populations include the size of the area, the road network, deer behavior or movement patterns, and legal restrictions. How immigration might change as densities decline is often not known. Now, the first aerial image here on the left is of Fairfax, Virginia, which would be a fairly good site because most of the area is evenly crisscrossed by roads. The second image on the right is in an area of Cincinnati, Ohio, and this would not be a good site because there are large areas that are not easily accessible from the road. And these park-like regions can serve as refuges for deer and limit access. Expanding the view beyond the area where deer will be treated is important because nearby populations may simply move in and fill the void left by deer that were removed. Deer are able to swim across rivers, so such bodies of water usually can't serve as physical barriers to immigration. Understanding something about the animal's movements and factors affecting survival rates is also important so that contraception can be strategically implemented. For example, mule deer in Oregon have higher survival rates if they migrate to other areas during the winter compared to those that remain in one location. So baiting in the winter may be more effective when targeting resident deer populations for contraception. And it takes time to realize the benefits of contraception, especially for animals like horses that only breed once a year and have relatively long gestation periods of 11 to 12 months. Removing animals and then contracepting remaining animals can more rapidly bring down population numbers. For horses, removals that result in animals being either adopted or placed in long-term holding should be coupled with contraception to maintain population numbers. For deer, legal hunting can control populations, but again, this is not usually a popular option in suburban areas. When population numbers are high, some combination of removals and contraception is usually most effective. Females are usually targeted for reproductive control because one male can inseminate many females. There are three broad categories of fertility control, including steroid hormones, sterilization techniques, or immunocontraception. I'm only going to be focusing on immunocontraception because there's no concern of anything being passed through the food chain. Vaccinations are minimally invasive, and to date, most vaccines are reversible. Immunocontraception works by stimulating the body to produce antibodies that can target gamete production, like gonadotropin-releasing hormone, maturation, like oocyte growth factors, function, which is what PZP or porcine zona pellucida vaccines do, and that's what I'll be focusing on the rest of the talk. And antibodies may also affect gamete outcome, like maternal recognition factors. Contraceptive vaccines are developed with an antigen, which is one of the targets I just described, and an adjuvant like Freund's complete adjuvant, modified Freund's adjuvant, or Adjuvac, to stimulate the immune system, and then other substances like liposomes or immune stimulants. Considerations include safety for the animals being treated. In general, the more targeted and downstream the treatment is, the safer the vaccine will be. So antibodies that work at the level of the ovary are less likely to have systemic side effects. For example, serum concentrations of GnRH normally start increasing at the onset of puberty. And when a juvenile male elephant was vaccinated with GnRH, stunted growth resulted. Requirements for delivery method and boosters, as well as vaccine efficacy and duration, will contribute to the overall costs and logistics of treatment. For the remainder of this talk, I'll be focusing on PZP vaccines. PZP vaccines result in the production of antibodies that target the ovary and bind the zona pellucida, shown here, that surrounds each egg to prevent sperm from binding and fertilization from happening. Zonostat was the first PZP vaccine developed in the mid-1980s and has been used in a variety of animals because ZP glycoproteins are well conserved across mammalian species. Zonostat requires a booster 4 to 6 weeks after initial vaccination and annually thereafter to maintain antibody titers. PZP22 was first developed in the early 1990s and over time has encapsulated PZP in a variety of microspheres and mini pellets to extend efficacy. And this was successful up to 22 months post-treatment in horses. Spayvac was also first tested in the 1990s and uses a special liposome technology to provide extended efficacy, which averages 4 to 5 years across deer species and horses with a single injection. Which PZP vaccine is quote unquote best really depends on the specific population and goals. For example, contraception that only lasts for 1 year may be preferable over longer lasting fertility control if the animals are easy to access and the ability to control reproduction of specific females is desired. However, for many free-ranging wildlife populations, multi-year or permanent contraception conferred with a single injection is preferred. Liposomes are multi-layered concentric spheres made up of phospholipid bilayers separated by aqueous compartments and they may themselves be considered immunological adjuvants. Sandwiched between the layers are the adjuvant and antigen, which slowly get released to the immune system. This results in a long-lasting multi-year immune response in infertility. The unique liposome technology was developed by IMV, a company that's based in Nova Scotia, Canada, and they currently use this platform for human vaccines. Spayvac for Wildlife has an exclusive licensing agreement with IMV to use their liposome technology in free-ranging wildlife and feral animals. Spayvac comes in 2 formulations, VaxiMax and DPX. VaxiMax is the original IMV formulation that needs to remain frozen until shortly prior to use. DPX is a freeze-dried version of VaxiMax, and it's mixed in an oil-based diluent just prior to use. It might be easier to use in the field because it doesn't have to be kept frozen prior to use. So now I'd like to talk about some of the trials with Spayvac. The first trial was initiated in the early 1990s on Sable Island off Canada's east coast. Gray seals haul out there every year where they give birth, nurse their pups, mate, and return to the sea. And all of this happens in just a couple of weeks. 101 female seals were treated with a single injection of Spayvac, consisting of 100 microgram PZP in Freud's complete adjuvant in the VaxiMax formulation. All of the treated seals had just given birth and were therefore proven breeders. But because the seals were inoculated just 1 to 2 weeks before mating, there would not have been enough time for PZP antibodies titers to fully develop, and no significant reduction in fertility was expected during the first year post-treatment. Normally, 25 to 30% of the seals that breed in any given year will not breed in the next. And you can see that was the case for the controls that had fertility of 70 to 75% for 5 to 10 years post. During this time, fertility rates in Spayvac treated animals ranged from 9 to 15%, and this was an 80 to 85% decrease. Results during the first year post-treatment were intriguing because only 47% of the treated females had offspring, when no effect was expected. In seals, development of the fertilized ovum is arrested at the blastocyst stage, and it later implants in the uterus. So it's possible that the PZP antibodies that did exist attached to the ZP, which surrounds the blastocyst, and interfered with implantation. The first Spayvac trial in deer was conducted on James Island in British Columbia, Canada, with free-ranging fallow deer. 41 does were vaccinated with Spayvac, consisting of 100 microgram PZP with Freud's complete adjuvant in the VaxiMax formulation. The does were followed for up to 3 years post-treatment. However, due to accessibility challenges, only 19 animals in the first year, 10 in the second, and 5 of the 41 originally vaccinated does were identified during those years. But none of them had become pregnant, compared to 96% of the treated or the untreated animals that served as controls. Here's a more detailed chart and additional results from 3 Spayvac trials in 2 species of deer. Not all of the free-ranging deer in British Columbia and Texas could be relocated, and the number that were identified each year is provided here in parentheses. 32 white-tailed does in Texas were vaccinated with 200 microgram PZP with Adjuvac in a MaxiVax formulation. None of the treated animals spawned during the first 2 years post-treatment, compared to 78% of the controls. And 1 of the 8 does identified from the original 32 treated fawned in year 3, compared to 100% of the controls. The trial in Pennsylvania was done in captivity, and both VaxiMax and DPX formulations were used in vaccines made with 200 microgram PZP and Adjuvac. The VaxiMax trial was actually carried out to year 5, and during years 4 and 5, 1 doe in the VaxiMax group fawned. The DPX trial went for 7 years, and 1 doe fawned each year from year 3 through 7. So contraceptive efficacy remained at about 80% for 7 years in that population. It's likely that the high plane of nutrition in these captive does boosted the fertility rates in controls and also made a robust immune response possible in the treated animals. A preliminary trial to see if Spayvac could raise antibody titers in African elephants was also completed in the US. 6 female African elephants were vaccinated with Spayvac using 600 microgram PZP with MFA. 3 received the VaxiMax formulation, and the other 3 received the DPX formulation. Vaccines were administered by IM injection in gluteal muscles, and blood samples were collected weekly for 8 weeks, then every 2 weeks for the next 8 weeks, and then finally monthly for an additional 5 months during the first year. And then antibody titers were monitored annually for up to 7 years. This graph illustrates log PZP antibody titers through time for the DPX formulation. Titers were first detected after 4 weeks post-vaccination, but they didn't peak until the first year. After that, they remained consistently elevated through the end of year 7. Elephants vaccinated with the VaxiMax formulation took longer to develop their antibody titers, anywhere from 2 to 9 months is when they peaked. And unfortunately, most samples 1 year post-vaccination were not collected. So we're just showing DPX results. Additional research is needed to determine actual contraceptive efficacy. And we will soon be measuring antibody titers for 3 of the elephants that were in this study, which will give us results for antibody titers up to 9, 12, and 16 years post-vaccination. 3 separate trials have been conducted in horses using a variety of Spayvac formulations and adjuvants. The first trial was conducted with semi-feral horses in Nevada. 12 mares were vaccinated in the neck using 400 microgram PZP with Adjuvac in the VaxiMax formulation. Killian et al. Observed pregnancy rates ranging from 0 to 17% for 4 years after treatment with a single injection. In comparison, 8 untreated mares had pregnancy rates ranging from 75 to 100%. So these were pretty encouraging results. Another study was conducted at Oregon State University with Spayvac using 200 microgram PZP with MFA. 7 mares received the VaxiMax formulation and 7 mares received the DPX formulation. All mares were vaccinated in the neck in early March, and the study ended in late October or late September, early October. We collected blood at 1-week intervals for 7 months to determine serum concentrations of progesterone, as well as PZP antibody titers, and we performed monthly transrectal ultrasound to image reproductive organs and record ovarian changes during the estrus cycle. At the end of the study, we ovarectomized 4 mares from each experimental group, so 12 mares in total, including controls, to obtain ovaries for gross and histopathological examination. Some of our findings surprised us. By using ultrasound and ovarectomies for direct examination, PZP vaccine was shown to affect ovarian function in horses for the first time. Treated mares had lower serum concentrations of progesterone, smaller ovaries based on size and weight, and had fewer follicles in comparison to control mares, all significantly so. The image in the upper right here demonstrates immunohistochemistry staining of the zona pellucida of a mare's follicle that was treated with immune serum. In 2017, this study was repeated by June et al. In a 6-month trial with pony mares that were vaccinated with Zonistat and boosted at 5 weeks. So there may be additional mechanisms of action at work in horses vaccinated with PZP. The ZP is thought to play a role in granulosa cell differentiation and folliculogenesis, so PZP antibody may be disrupting cellular communication between differentiating follicular cells and the developing oocyte, which may lead to the destruction of a portion or all of the oocyte pool. Subsequent 3rd trial was initiated with horses removed from rangelands and held in long-term holding facilities in Oklahoma. 60 mares were vaccinated with Spayvac using 200 microgram PZP with MFA, and 30 received the VaxiMax formulation and 30 received the DPX formulation. And all mares were vaccinated in the rump. Blood samples were collected prior to vaccination and then monthly through the breeding season, when proven stallions were rotated in every 2 weeks. Fertility was determined through hormone assays, rectal palpation and ultrasound, and direct observation of foals. And a female was considered fertile if any one of the 3 measures was positive. Results were again somewhat surprising. The DPX treated mares had a 17% fertility rate during year 1 compared to 100% for controls, but this jumped to 76% during year 2, and this group was removed from the study. So this graph shows results for the VaxiMax treated mares, and they had fertility rates ranging from 13 to 47% for the first 4 years compared to 83 to 100% for controls. And then this climbed again in year 5. Investigators found that 5 of 30 VaxiMax treated females were not cycling for more than 3 years after vaccination, which may suggest that loss of ovarian function can be permanent when it occurs. However, overall, these results were not as robust as expected, given findings from the previous 2 studies. So what happened? There were some differences in the amount of PZP and the type of adjuvant used, but this would not likely have an effect. The Oregon trial used 200 microgram in MFA like Roelli did in Oklahoma. In the 2 studies with the most robust results, vaccinations were given in the neck, while the Roelli study, vaccinations were given in the rump, specifically the superficial gluteal muscles up here. Could that make a difference? As it turns out, yes, we believe it could. In several other species, injection site has made a difference. For example, in humans, several studies have shown that the immunogenicity of a vaccine is higher after IM injection in deltoid arm muscles than in thigh or gluteal muscles. The dermis contains dendritic cells, which facilitate the capture of antigens, and local inflammation induces maturation of these cells and their migration into draining lymph nodes. Lymph nodes serve as filters for the body and contain immune cells that respond to foreign substances like PZP and start producing antibodies. This figure illustrates some of the regional draining lymph node areas in the horse, and these are some of the major lymph node sites. Injections given in the neck of the horse are virtually on top of a major lymph node cluster, whereas injections in the rump are further from the lymph node cluster. So site of injection in a large animal like a horse may play a role in Spivak's efficacy. The Roelli study also noted that consistently infertile mares, which were 15 of the 30 treated VaxiMax mares, didn't necessarily always have the highest antibody titers. So in another study, we looked at serum samples collected 1, 2, and 4 years post-treatment from 14 treated mares that were consistently infertile compared to 14 treated mares that had foaled at least once during this time period. IgG is the predominant antibody-producing immunoglobulin, but the horse also has 7 IgG isotypes, which we're learning have more targeted roles. We measured IgG isotypes in serum samples from these 2 fertile and infertile groups from the Roelli study using a fluorescent bead assay. The top graph depicts differences in antibody titers between fertile and infertile treated mares as measured by ELISA. And as you can see in the lower graph, the fluorescent bead assay was more sensitive in differentiating effectively contracepted mares than the standard ELISA antibody test. Interestingly, the mares that were consistently infertile for 4 years had significantly higher levels of IgG47 isotypes than mares who'd foaled at least once. IgG1 and 47 antibodies are typically involved in responses to viral infection or intracellular bacteria, while IgG3 and 5 isotypes are induced after nematode infection or during allergic responses in the horse. Also interesting, additional blood samples were collected from these 14 consistently infertile mares during year 5, and their levels of IgG47 titers were significantly higher compared to levels during year 4, and there was no booster involved. So clearly we have much more to learn about all of this, but IgG47 could serve as an indicator of effectively contracepted mares. So dendritic cells mentioned earlier link the innate and adaptive immune systems. They are very cool cells, in my opinion. They are abundant at body surfaces and within tissues where they sense and sample the environment for self and non-self antigens. Once activated, they migrate to the lymph nodes where they interact with T cells and B cells to initiate and shape the adaptive immune response. Their main function is to capture, process, and present antigens to adaptive immune cells, which include B lymphocytes responsible for antibody production and T lymphocytes responsible for cell-mediated immune responses. Different ways to improve vaccine efficacy usually involve dendritic cells. Some proposed mechanisms for maintenance of PZP antibody titers include Spivak's liposome matrix, which provides sustained release of antigen, and this may lead to enhanced recruitment of dendritic cells. The antigens may induce a strong T cell response that initiates mobilization and maturation of dendritic cells, which migrate into the injection site area. In fact, many of the mares in both the Oregon and Oklahoma studies had varying degrees of an inflammatory reaction at the injection site that waxed and waned over several months. These various initial trials with Spivak raised additional basic research questions. For example, can PZP antibody titers also interfere with blastocyst implantation in seals? Does Spivak's ability to preferentially stimulate production of IgG47 isotype contribute to its long-term immunocontraceptive efficacy in horses? Does something similar occur in other species? Does injection site affect immunocontraceptive efficacy in the horse and perhaps other species? How does health status, which can fluctuate by season, affect vaccine efficacy? We've never tried giving a booster of Spivak. Would it have a synergistic effect in extending contraceptive efficacy or render females permanently infertile? We're also interested in seeing how Spivak works in other species or the formulation used with other non-PZP antigens. We have a few studies currently underway and are waiting for preliminary results. 50 free-ranging macaques have been vaccinated, 12 free-ranging red deer, another 8 will be vaccinated this winter. 1 captive white-bearded and 2 white-handed gibbons were vaccinated. Future trials are also being planned, studies with horses to test differences in contraceptive efficacy based on neck versus rump injection sites, VaxiMax versus DPX formulations, and booster effects. An antibody titer study with captive Asian elephants is being planned and will hopefully be followed by contraceptive efficacy trials in free-ranging populations. And a study to test the contraceptive efficacy of Spivak and nilgai might also be a possibility. And then there are other species which may require different antigens. They include feral dogs and cats, pigs, and even cattle. So clearly a lot of work remains. I know I covered a lot of material in this talk, but I hope it was of interest and helpful. You can visit the Spivak for Wildlife website for more information as well. So I'm happy to take any questions.
Thanks. So Dr. Sambasiva Rao has raised hand, so maybe you can unmute and speak. Dr. Rao. Well, we have a question here from Parag Dega. He wants to know, will it able to use in pig?
What was the question?
So this, yeah, so he is
unmuted. Can I?
Yeah, sure. Go ahead.
Yes. Ursula, I was in touch with the Creek Patriarch at the initially in around 2000 about use of PZP in the pygmy hawk, and then he was saying me that at the time it's not possible because it's a pig. So he said, so better to use other options. But not now you show that you were showing that it can be also tried in pig because it's the same species, swine.
Yeah, I think it's probably easier to use a different antigen, but if you can stimulate the immune response to respond to that, I don't see why it wouldn't work, I guess. But you've heard some success?
No, because Craig Patrick was saying me that it's not possible to use for pig. You better use other agents. So I use Depo-Provera for pygmy hog, and it really works for some time.
Okay, what was it that you used?
Medroxyprogesterone.
Oh, okay.
Okay. But I contacted him initially around year 2000, soon after they developed the PZP. It was at the trial stage at the time.
Yeah, there are a lot of different options, different ways to formulate vaccines.
Yes, I learned now, yes, from your lecture. Thank you.
So one more question is there from Dr. Narayan. So he wants to know that this tool becomes politicized. So just like that, what happened to climate change topic? So could that be a worry regarding ownership? Your thoughts on that?
Whether this could be politicized?
Yeah. And then.
Oh, I think for certain species, it definitely is. I know in the US, the horse population, there are a lot of very opinionated groups involved. But I do belong to a coalition that is working across groups, trying to communicate with them, but it's still challenging because everybody has an opinion, and so there's no easy way forward when it gets emotional like that. And I think whenever you're working with a charismatic species, it can be that way. And I think social media can really contribute to the spread of misinformation, which isn't very helpful.
Thanks a lot. Thanks for the wonderful talk, Dr. Ursula. And now let me invite Dr. Umapati to introduce last speaker of the session, perhaps for the day, Dr. Douglas. So Dr. Umapati, please take over. Thanks, Ursula. Yeah, great.
Thank you, Ursula, for your wonderful talk, a very explanatory talk. People may be interested in controlling population. Thank you so much.
Thank you.
Good evening, good night, and good evening, good morning once again. And I'm happy to introduce the final talk by Dr. Douglas Ekri. Douglas Ekri is an assistant director for the USDA APS Wildlife Services National Wildlife Research Center, NWRC, in Fort Collins, Colorado, USA. In this role, he oversees several research projects that focus on development of methods to reduce human-wildlife conflicts while preserving the environment. Prior to this position, he was a leader of the fertility control project at the NWRC. Research interests are reproductive biology, biotechnology. The research has been focused on ovarian follicular growth, pituitary function, and on development of reagents for fertility control of wildlife and invasive species. I may welcome once again Douglas to take over from here. Hi.
Very good. All right. Thank you. Good. Thank you. Nice to see and hear you.
Yeah.
Let me try to share my screen. Start the show. All right. Okay, I think we're good. Thank you very much, Dr. Umapati. It is nice to see you, and thank you to you and to all of the organizers of this conference for inviting me to attend and to participate in this. It's a real pleasure. So as Dr. Umapati said, I do work for the USDA National Wildlife Research Center. It's located in Fort Collins, Colorado. Our mission is to apply our scientific
Just a second,
expertise.
Dr. Douglas. Can you make it
Yeah.
presentation mode? Just your screen is in still not in a presentation mode. Your screen, so you can just make it.
Oh, okay. So I need to change. Let's see. Is that better?
No, still.
No. Okay.
Put on the slide mode, you press this button there. So,
Yeah,
Yeah.
yeah.
yeah, it's good.
Has that worked?
Yes,
Yes. Very good.
Okay, good. Thank you. All right. So I've got too many screens, I guess. Anyway, so as I was saying, our mission is to apply our scientific expertise to resolve a variety of human-wildlife conflicts here in the United States and throughout the world. And we want to do that in a way that will maintain the quality of the environment that we do share with wildlife. That's an important aspect of what we do. So the talk today is about fertility control. We've heard some about it, and there'll be another talk after this as well, I believe tomorrow, about fertility control. So we asked the question, why would we want to do this? And so this is an idealized graph of fertility control, where we start with a population of animals, a certain number. And in the perfect situation, we would be able to reduce the number of animals in that population by whatever percentage. We know that without further control, that population of animals will be able to recover and increase again, and usually get to the same level it was before. The idea is that we can then combine that with fertility control once we've reduced the population, and that population growth again will be slowed and maybe even just stopped. But this line, this horizontal line, this dotted line here, represents just a threshold. And that can be whatever the threshold is decided. And that would depend on the objective of the effort, whether that's for some sort of conservation goal. It might be for disease management. It might be towards an eradication of an introduced invasive species. And so whatever that threshold is, that needs to be decided by whoever is performing the management regime. And so in this idea, so the advantages then of fertility control would be, one, reduced frequency and cost of control if we combine that. And it's important to point out in this that we have the conventional and fertility control. It's important to remember that an integrated approach is going to be very important. There isn't a single tool generally that is going to create the solution to whatever it is that we're trying to achieve. So oftentimes, for many species, there are poisons that are used to manage a population. And so the use of fertility control would certainly reduce the amount of poisons that might be used and preserve the environment as well. It could have reduced risks to any non-target species as well. One of the goals is to have something species specific. That's not always possible, but it would certainly reduce the risks to them because it's non-lethal. We can increase the scale of the management effort. Sometimes it's just small populations that are being targeted, but often we want to treat a population or populations at a landscape scale, and this might afford that ability. And then because it is non-lethal, it is often regarded as being more humane to the target species. And so, as we know, fertility control, like most things that we're doing, whether it's for conservation and all of these reproductive technologies, it's complicated. And so in developing a fertility control, we need to first identify whatever physiological mechanism or the molecular targets of reproduction that we want to target. And those could involve hormones, those could involve fertilization, it could be growth factors. There's a variety of different things that could be targeted. We then need to develop a reagent or some process that will, in fact, inhibit whatever that target is, that physiological target. We then need to go to the next step of delivery, which is a huge challenge, actually. So how do we deliver this reagent that we've developed in the lab and be able to deliver it to the target species that we have in mind? And there's a lot of things to consider in delivery, not just the gut physiology when we deliver it, immunology if it's a vaccine, the formulation, the manufacture, all of these things go into that delivery piece. There's also whether you want to target the immune system or utilize the immune system through immunocontraception, or is it going to be something that's going to be what I call direct acting? In other words, just not utilizing the immune system. And then there's another level that it could be a non-transmissible system, say a bait that we would put out on the landscape, or we have explored and investigated different options of a transmissible system. So in Australia, for instance, for many years, they were looking at virally vectored immunocontraception. And when I spent some time in New Zealand, we were looking at using a gut nematode as a transmissible system of delivering a fertility control. The ideal system would be to be able to put up a sign and ask all the animals to come to the clinic and get their shot or get their treatment. Of course, that's not possible. And so we're left with the challenges and those very large challenges that we have. So we combine those two things of having our reagent and a method of delivery, and then we hopefully have a potential system of delivering an effective fertility control. But even when we get to that stage, there's a lot of modeling that would be involved in how we might best apply it. There's the social science and the ethics portion. And then, of course, there's the regulatory aspect as well. All need to be addressed and all are challenges in their own right. But at the end of this, and you see with the arrows going around, this is a very iterative process. And as we learn more, hopefully that we can improve the things that we're doing. And ultimately, we want to come out of this some new management tools. And as I mentioned, there isn't going to be a single tool that's going to be effective for every situation. And Ursula made the point very well that there are a variety of tools, and we need to choose those tools based on the objectives that we have when we're trying to manage those populations. And so within the National Wildlife Research Center, the NWRC, we have a fertility control project, and it's currently led by Dr. Jason Bremer. It used to be me, and we've now got Dr. Bremer on board, and we're very pleased to have him. And so, of course, our goal is to develop these reagents and methods that would be effective, suitable for practical delivery, and meet the need of wildlife managers. But I would also add in there's that social science aspect, the human dimension. So it needs to be acceptable to a very large number of people. Our motto, if you will, is one-hit permanent sterility. For many of the species that we work with, we're happy to be able to treat them once. To be able to treat them more than once would be an even larger challenge. And so we are looking for something that could be delivered as a single time and cause permanent sterility for many of the species that we're interested in. And so we have a number of research areas that we're interested in. One, and I'll touch on this in just a little bit, is oocyte depletion. So depleting the ovaries of all the eggs that are present, and which would then result in permanent sterility. We also have been and are still involved in vaccine delivery or development. So we have the injectable vaccine Gonacon, and I'll talk a little bit about that in a moment as well. We've done some research in mucosal or oral vaccines, which would certainly have advantages to delivery in some senses, and then looking for new or better target antigens that might afford some of the things that we're after, in particular permanent sterility. And so in doing this, though, we need to keep in mind some of the criteria that have been put out or mentioned for what a fertility control reagent should be. And the top one, at least 90% effective. And so that, I would say, would be in a very controlled situation. If we're not at least 90%, then we need to try more, because we know once we get out into the field that that's going to decrease for a number of reasons, whether it be delivery or just the way the animals in the wild are slightly different than those that are in captivity. And Ursula made a good point again about those in captivity are generally in very good health because they are in captivity, and they might be able to respond to some of the treatments that we give just a little bit better. It should be amenable to remote delivery, and that's certainly if we want to be able to treat at a landscape scale. Some would argue reversibility. Now, I said that our motto is permanent sterility, and so that's something that needs to be considered, again, depending on the objectives of what you want to do and how you want to manage that population of animals. It needs to be safe for pregnant animals, no significant health or behavioral effects. It doesn't enter the food chain for certain species, say if it was for deer or something like that. Again, minimal effects on the individual and social behaviors. And the one that everybody's interested in, that bottom line, it needs to be cost effective. So it can't cost a whole lot either. And then I put in there a different color bullet on species specificity. And so that could be in a couple of ways. It could be for the reagent itself, which would be ideal. But often many of the reagents that we have or methods we have currently aren't species specific themselves, but we can offer some species specificity in the way that those things are delivered. And so we look at that aspect as well. So what are some of the current tools that are available? Of course, we have surgery. The surgical methods, ovariectomy, the ligations, IUDs, and vasectomies, and they are very effective. In cold steel, it's hard to beat for the permanence, but some of those other criteria that I mentioned, it might not hit the mark as well.
You're muted, Dr. Douglas. Please unmute and speak.
Oh, I am muted.
Muted.
There we go. Okay, sorry about that. So we have a number of pharmaceutical agents also that can be used. There's progestins, estrogens that could be put in foods or also as implants. We have hormone agonists. And the photo on the right side there is an agonist called desilurelin. It's a GnRH agonist that has been used in certain species. Prostaglandins, there have been some intratesticular agents that have been tried, and chemostimulants are being developed, although I don't know that we have any 100% chemostimulants at the moment. And then we have vaccines that we've heard about. Gonacon, the gonadotrophin releasing hormone vaccine, and the porcine zona pellucida, or the PZP. And I'll be talking about a few of these as we go along. So at the moment, there's only a handful of things that are registered for use in the United States. And they're registered as pesticides. And there's a story behind that. If anybody's interested, I can talk about that. But at the moment, those that are registered for fertility control in wildlife are registered as pesticides. So there are a couple of vaccines. There's the Zonistat H and D, which is either for horses or burros and deer, and Gonacon, which is also registered for deer, horses, and burros. There's a chemical reagent called Contrapest, which is the active ingredients are VCD, or 4-vinyl chlorhexidine diapoxide, and triptolide, and that's for rats. And then there's a reproductive inhibitor, ovocontrol, which is nicarbazine, which is registered for pigeons. So like I say, there's only a handful that are actually registered, although there are several others that are used in more research sense. And so we've already talked about immunocontraception. It essentially tricks the immune system into treating a target reproductive protein as foreign. And so that's a trick in itself. And it's amazing that we've been able to do that, and that it has also been effective, and sometimes by a single injection. And so that, again, the two vaccines that are available now, at least registered vaccines target the PZP, the zona pellucida, Zonostat H, and then Ursula just talked about Spayvac, which is also available, and then Gonacon, which targets gonadotropin releasing hormone. So Gonacon was developed by scientists at the NWRC. It was the first contraceptive vaccine registered for use in wildlife. It was registered by the Environmental Protection Agency, or the EPA, and for white-tailed deer, female white-tailed deer in 2009, and then for female feral horses and burros in 2013. The vaccine, we have licensed that to a company called Stay First now, and so they are now able also to manufacture and distribute that vaccine for operational uses. So what is Gonacon? Well, in order for it to work, GnRH itself is a very small peptide protein, only 10 amino acids. And so it needs to be linked to a carrier protein. And then for Gonacon, the GnRH is linked to either KLH or blue protein, it's called. And these are mollusk hemocyanins, which are they're large, but they're also seen as a foreign protein to a mammalian immune system. And so as many GnRH molecules are linked to these carrier molecules, and then it is mixed with a mineral oil-based adjuvant, in this case, Adjuvac. And a key ingredient in Adjuvac is killed Mycobacterium avium. And so many of the adjuvants, the oil-based adjuvants, have a Mycobacterium component to it. The Adjuvac happens to have Mycobacterium avium. And so these are all combined and they're made into an emulsion, and then it's provided in preloaded syringes with the vaccine in it, with the volume based on whatever species is being targeted. So although it's only registered for use in deer and horses, it has been tested and found effective in a whole variety of other species. And so we've got work all over the world where we've been testing and trying to find out how useful it is in a variety of other species, from like I say, deer, horses. You can see bison there, feral pigs, or wild boar in other places like Hong Kong, and a number of marsupial species. It's been found to be very effective. In fact, in the kangaroos, the eastern gray kangaroos, a single injection has been shown to be effective for about 10 years. And so I wanted to focus a little bit on some of the work that we've been doing with horses. That's a species of interest. It's very important here in the United States that the feral or wild horse populations are managed. And Ursula went into that and gave a nice background to that. So back in 2009, we were able to start a study at Theodore Roosevelt National Park in North Dakota. And so a number of 25 mares were treated with Gonacon, a single injection, and there was an equal number of control mares as well. And as you can see, when the vaccine was given, the fertility was essentially the same for both the control and the treated animals. A year later, it was the same because they were mostly pregnant when they were treated, and so they all foaled. But then the next 2 years, although it was modest, there was a reduction in fertility in those animals. In 2013, the park was going to manage those horses again. And so we had the opportunity to go back and retreat those mares with a booster dose of Gonacon. And as you can see in the subsequent years, that following the booster, the vaccine was very effective at reducing fertility in those mares. And so that's not a huge surprise. We know that boosters are effective, but we were very encouraged by the length or the duration of efficacy after that booster in those mares. And so some of the take-home messages from this was, one, that the booster is very effective over multiple years. Importantly, it's safe in pregnant animals. And also, very importantly, some of those criteria, there were no negative effects on the social behaviors or the herd structure. So those bands stayed together as they would normally. So that was nice that we were able to bring these animals in. They were all rounded up. And so we were able to run them through a chute and treat these animals by hand injection. And so in this table here, we see the hand injection, the booster interval for those was 4 years, and the efficacy was very good. We wanted to look at a maybe more practical method of delivery, because we can't always round them up. And sometimes we do need to treat animals by remote darting. And so we are currently looking at 3 different intervals of that boost, a half a year, 1 year, or 2 years. And you can see currently over 4 or 3 years, the reduction in foaling is about 60%. And so it is effective. Interestingly, not as effective as by hand injection, at least for this data, but it is effective. And you can see that it's certainly not permanent. We knew that, and we've just put some numbers to that. And so our current focus is on optimizing the use of this tool in the field. So we know it works. We now have some more parameters of how it works and what to expect. And so now we want to help people who might be interested in GonaCon at how to utilize it best. What would be the best interval based on how they can treat the animals, whether they can round them up or whether they have to use the remote darting or whatever that might be. But the results are very encouraging, and it's just another tool. Like I say, this is one method, and where those might be interested, we're certainly happy to help them understand how best to utilize this tool. And so now I want to change gears a little bit and talk a little bit about the work towards permanent sterility. So GnRH is good, GonaCon is good, but it isn't permanent. And so we're targeting the population of eggs in the ovaries. We know that mammals are born with a finite supply of eggs, and they're found in primordial follicles. And every day, a certain number of those follicles begins to grow. And then over a certain amount of time, depending on the species, one or multiple follicles matures, and then ovulation occurs. Most of those follicles die along the way. So less than 0.1% of any of the eggs ever go on to ovulate. The vaccines that we currently have available, the GnRH or ZP targeted ones, impact towards the end, either that final maturation or ovulation or even fertilization is where they are. So yeah, they are effective and they can certainly target these things effectively. But as we know, there's still this supply of primordial follicles, hundreds of thousands of them, in fact, that are just going to keep replenishing that supply. And so we need to be mindful of that. And so the question is then, how can we cause oocyte depletion? Well, we know that because the initiation of growth is a committed step. Once they start to grow, they can't stop. And most of them are going to die. So we could potentially just cause a mass activation of all those primordial follicles to deplete them all. Or we could come up with a reagent that would directly kill those primordial oocytes and or the supporting cells around them. So when I was in New Zealand, there was a line of research that was being conducted around fertility, trying to increase the fertility of sheep, trying to increase lamb production. There was a very clever scientist named George Davis down at the Invermay Research Center that was tracking the genetics of certain strains or lines of sheep. And there was one that was called the Inverdale sheep, and it had a very interesting phenotype. And so in these animals, if they were wild type, they had an ovulation rate of maybe 1 or 2, just the normal ovulation rate. If they had a single copy of a mutation, then it actually increased the ovulation rate. So that would increase the lamb production by 1. However, if they had a double copy of that mutation, they were completely sterile. And so they knew about this strain for many, many years, but they really just didn't know, or we didn't know, what was the cause of it. And so we did a lot of endocrinology and all sorts of different things. But then as the tools became more available for the molecular biology, it was possible for us to identify what was causing this mutation, what were the genes involved. And it turns out that there are two oocyte-derived growth factors, one being growth and differentiation factor 9, or GDF9, and the other is bone morphogenetic protein 15, or BMP15. Interesting names, because they are oocyte-specific and oocyte-derived, and to have been given those names initially is interesting. But they are indeed oocyte-specific growth factors that are involved, and they were the cause of this mutation in this Inverdale sheep. As we've learned more about these growth factors, we found that they do have very species-specific actions, whereas in a sheep, mutations in either of these will cause complete sterility. In other species, one or the other would cause the same things, and the impacts are very different. And we have done some research in some other species. In my case, we've treated deer with either GDF9 or BMP15 and found that they do impact fertility in that species as well. But again, we're interested in horses at the moment. And so I thought I'd just quickly go through some of the results that we've had when we've treated horses with either GDF9 or BMP15. So through the molecular world, we did know that the oocytes of horses did express both of these oocyte-derived proteins. So we immunized horses against one or the other, GDF9 or BMP15. And as you can see, there were some impacts, but they weren't very profound. They weren't the impacts that we were looking for as far as developing a vaccine or otherwise for a fertility control reagent. Fortunately, we were able to maintain some funding and continue the research and immunize horses against a combination of both GDF9 and BMP15. And that's where we saw the very profound effects, the ones that we were looking for. And so when we treated animals with these growth factor, the combination of them, there were no ovulations that resulted. In fact, follicular growth was inhibited. There were no follicles greater than 12 millimeters that were found on the ovaries, and this was monitored through ultrasound. In a horse, a mature follicle that's ready for ovulation gets up to 40 or 50 millimeters in diameter. And so to be less than or equal to 12 millimeters, we did inhibit that growth. And there was no estrus in any of these animals. And in the graph below, you can see this is progesterone in those animals. The blue line, or the top line, is from the control animals, and you can see those animals cycled normally. But the red line at the bottom, that was from the treated animals. So they just weren't ovulating. So that was manifest by those progesterone profiles. And so that was very exciting to us because these animals were treated with what we would regard as a very mild adjuvant. And we did it in a very controlled way to maintain the antibodies that we wanted. We were again able to maintain those animals for an additional year where we didn't treat them. We just monitored them for another year, and we had essentially the same results. And so even without a booster, we had 2 years of essentially complete infertility in those animals. And so we're very encouraged by these results. We've kind of got the proof of principle that these growth factors are involved in horse reproduction, and there is some scope and so potential for these to be utilized as a fertility control reagent and maybe even a vaccine. And so our ongoing research is looking at adjuvants that might be used to create that single shot, the goal that we have of having that single shot that will cause either permanent or at least very long duration of infertility. And so we're looking at different adjuvants. We're looking at the duration after a single injection. We want to know whether it's safe to use in pregnant animals or not. There's no reason for us physiologically to think that it would impact pregnancy, but we do need to test that. And of course, we'll be looking at other species and the effects of these growth factors as well. And so again, I'll change gears a little bit. and touch on some of the work that we did with mucosal or oral vaccines. We know that injectable vaccines have some limited usefulness just because you have to target the animals and be able to inject them. Vaccine adjuvants often, as I mentioned, contain some species of mycobacterium. In Adjuvac, it's Mycobacterium avium. That's the mycobacterium that's utilized in that. So we asked the question, can M. avium be used as a mucosal vaccine? And one of the reasons why it's used in Gonicon is that M. avium is ubiquitous in the environment. And so most species, at least in the United States that we've tested, wildlife, have already been exposed to M. avium. And so when we utilize it in Gonicon, the idea is that it's seen more as a booster than the single or the first injection. And so that's part of the reason why it's so effective. But we wanted to know whether this could be used in a mucosal vaccine. And so in collaboration with some collaborators in the UK with Dr. Giovanna Massey and Dave Cowan and some others, we were able to create fragments of M. avium. And then we conjugated, in this case, it was a recombinant protein that contained the sequence for GnRH. It's called IMX294, and it's made by a company called Emaxio. And so we were able to conjugate these fragments of M. avium with this GnRH containing recombinant protein. And then we treated rats, in this case, they were lab rats, by oral lavage. So not gavage, but lavage, just put it into their mouths and let them swallow. And we had a number of different treatment groups. You can see where they had either 3 doses or 6 doses. We did it by injection as well. And there were 10 animals per group. And in the red rectangular box there, you can see where we got a little bit excited, and this is published in the paper that you can see below. But it was the first evidence where we could get an antibody response against GnRH when the vaccine was given orally. And it also reduced the proportion of females that had given birth. And so again, hugely encouraging. We have an awful long way to go with this, but it did, again, give us that confidence that it is indeed possible. We're not there yet. Like I say, we have a lot of work still to do in this area, but we're very encouraged that the potential does exist. Changing gears again, just another reagent that we've done some work with. This was a number of years ago, and we're reinitiating some of that research. It's a compound called diazepam, and it's a cholesterol inhibitor. And as you can see in the cartoon there, what the chemical is at the 20 and 25 positions, the methyl group is replaced by nitrogen. And so it acts as a cholesterol inhibitor. And we know that cholesterol is the base ingredient for all the steroid hormones that are made, including the reproductive steroid hormones. So it's been tested in a number of species. It's orally active, which gives it an advantage. And we found that it's effective in certain species of birds and in some small animals. And some of those that we've been interested in are the monk parakeet and the rose-ringed parakeet, which are considered pests in the United States and in Hawaii, and also in the gray squirrel and the black-tailed prairie dog. And in all these species, we found it to be effective. It's not permanent, so it's given to the animals several weeks before a breeding season, and most of these species are seasonal, and so that's important in the delivery as well. But it's fed to them over a 5 to 10 day period before the breeding season. The effect lasts throughout the breeding season. And then if you wanted to carry on, then it would have to be reapplied at the beginning of the next breeding season. But again, it works. It's orally active. There's still an awful lot of work to be done. For instance, we don't know what some of the non-target consequences might be. For instance, the black-tailed prairie dog is considered a keystone species, meaning that it's a primary prey species of other animals in the environment. In particular, the black-footed ferret. And so if an animal, if a prairie dog was to be treated and then consumed by a black-footed ferret, what would those consequences be, if there would be any? And so those are important questions that still need to be addressed if we were to, as we move forward with developing diazepam as a fertility control reagent for certain species. Some other areas that we're new, but we're getting into would be one, silencing genes with interfering RNA. This would be another kind of direct acting reagent. As we know, RNA is the template for protein production. And so with these interfering RNAs, we can inhibit translation of specific proteins. It would lend itself to some species specificity. And so we're excited to be going down this line of research. But again, it's very much in its infancy, but it is an area that we are actively involved in at the moment. Another is not so, maybe not directly regarded as fertility control, but gene drives. And this would be mostly to target introduced invasive rodents, say on island ecosystems. And so it's a genetic engineering technique. It's made so that the normal Mendelian rules of heredity are not followed. And so it increases the likelihood that a certain gene will be passed on to the next generation. You may have heard in Australia, they've been doing a lot of this for several years for the daughterless carp, the fish species. And so that would be the same idea for this in rodents, is that the progeny would be all males. And so they would essentially breed themselves out of existence in that local population. We're part of a big consortium called G-Bird, and there's a number of organizations involved in that. And we've done a number of several areas of research looking into this as a possibility. This is, again, in its infancy, and given that it would be something that transmissible out into the environment, we've got a long way to go before this is actually a reality. But it is an exciting area of research in which we're involved. So I'll end this by asking a question. Well, what is the ideal fertility control? And this idea came from a guy named Mark Fisher down in New Zealand. He was a social scientist who thought a lot about this. And one, well, it has to be technically feasible. And it also has to be cost effective, because everyone is concerned about the bottom line. What do things cost? But the second element is it has to be acceptable, not only to those who might utilize it, those managers, but also to the public at large. And there's a lot of different components of that public that we need to be mindful of. And so the ideal fertility control is whatever we propose as scientists, whatever it is that we develop, plus any justified differences, because there's never going to be one thing that's going to be 100% acceptable by everybody. And so there needs to be quite a dialogue as to what it is that makes it acceptable. And those are those justified differences. Ultimately, what everybody wants is something that's safe. And that's a difficult thing to address sometimes, because I would guess for as many people as are listening right now, there's probably that many definitions of what safe is. And so that's a tricky one. And that's where the social science and the human dimensions come in as we navigate the development of these different reagents for fertility control. And so finally, I'd just like to acknowledge there's a lot of people that have been involved in all of this research over several years. So I'm very grateful and indebted to a lot of these people for their collaborations and ideas, and certainly those who have provided funding for all of this research. And so with that, I thank you for your attention and listening, and I'd be very happy to address any questions if there are any. Thank you very much.
Thanks a lot, Dr. Douglas. And it was a nice presentation. We really learned how different tools, basically different tools are there to control fertility, and obviously there's different dimensions with respect to ethical issues with humans and all. So really nice talk. I'll take a couple of questions. I think Dr. Umapati has a question. He wants to know, is Gonacon effective against dogs and monkeys in a study?
So we've done some work in both of those species, limited, unfortunately, Gonacon, the registered formulation, while it may be effective, it does cause very severe injection site reactions, open abscesses and open sores. And so it's really not suitable for use in dogs. We have been working towards developing an adjuvant that is more suitable for dogs, but it's kind of finding that balance between something that really stimulates the immune system, but doesn't cause those injection site reactions. And so we haven't got it yet, but we certainly are working towards that. We've only done one small study in monkeys and vervet monkeys, and similar thing happened, that they had some pretty severe injection site reactions. So we don't have quite the right adjuvant yet for either of those species.
Thanks. Yes, Dr. Gupta, you want to?
Yeah, I think my question is partly already taken by Dr. Umapati. I also wanted to ask the same question, that whether the inclusion of Mycobacterium avium in EDUVAC will not lead to injection site reactions. And I think Douglas has answered that. My second question is, again, related to that, is when you are including Mycobacterium avium, which is already animal is exposed to it, and as you said that it may act as a booster, will the overproduction of antibodies against the Mycobacterium avium may compromise the antibody production against GnRH or other antigen?
Yeah, that's a great question. That's kind of, I don't know the answer to that.
Which we call as a carrier-mediated suppression, like in some of the short ligands or short peptides, which are conjugated with carrier, and once you generate the antibody against the carrier, it may suppress the immune response against the hapten or against the ligand, which may happen in this case also. In addition to the injection site reaction and the inclusion of Mycobacterium avium, an antibody response against it may also lead to some compromise on the antibody production if you simply use oil, water in oil emulsion without any mycobacteria.
Yes, so that's a great question. And we even had the same question around the carrier protein. So in the case of GnRH, we have to conjugate it to a very large carrier protein.
Yes.
And so we've wondered, have we moved that immune response preferentially to the carrier protein instead of the GnRH? And when we measure antibodies against, say, KLH, it's a very, very strong immune response. But fortunately, we don't know if there's a preferential response to it. Mm-hmm. We do know that there's sufficient response against GnRH also, that it is effective. And so, but that's a great question, whether we are doing something like that. Again, with the avium, we kind of need it in there as an immunostimulant to really get the immune system excited and to pay attention to our antigen. Where that balance is, I guess I don't know, but that's a really important point, and it's an interesting.
I think you are in a lucky situation because you have used only a single injection of Gonacon, and if you have to repeat the injections, then you will see this carrier-mediated antibody suppression.
Perhaps,
because, that's what we have seen in another vaccine meant for the women for contraception, where we find that you have to change the carrier in between when you are giving multiple injections. But luckily here it is a single injection, so I don't know how much it will affect.
Yeah, well, in horses, fortunately, the booster was very effective. We didn't change the carrier protein. It was the same exact formulation that we gave as a booster. So in that case, it worked out just fine. But you're right. I think, with the immune system, there's a huge black box that we still don't understand. And there's a very high amount of species variability in that black box that we don't quite understand. Sure. And you're right. Sometimes we're lucky, right? We try it and it kind of works out for us, and we're glad for that. Right. Serendipity is a good thing. Yeah.
Thank you so much.
You bet.
Thanks a lot. There's another question from Pratibha, whether gonacon has been tried in male contraception in human. And she also mentioned that BMP results were interesting in homozygous knockout. Further, she mentioned, I'm not sure, but still now there's no efficacy male contraception in human.
Yeah, so I don't do anything in human research. We've not provided gonacon to anybody who's doing any human research. So it is effective in males because it's GnRH. So we have tried it in different animal species in males, and it will work. Interesting story, we provided some vaccine to some colleagues in Nepal who were going to do a dog study. And that's where we found, again, that the gonacon itself has those bad injection site reactions. But some of the women of the village where they did ask that same question. Hey, would this be working in male humans as well? But I don't know, because it's never been tried.
Great. Yes, Dr. Gupta, you have another question.
I have one more question. You gave very good results with respect to GDF9 and BMP15 immunization in horses. If I remember correctly, I attended some international meetings, and the data which was presented there with both GDF9 and BMP15, perhaps in sheep or some other animal species, it did not lead to any contraceptive effect. Can you throw some light on that? Can it be a very specific effect, or if I'm wrong in my assessment? Yeah.
Yeah. So the research that all came out of New Zealand when I was down there when they were trying to do that. And so, in fact, the peptides that we're using now in the horses were based on work that was done in New Zealand, where they did the epitope mapping, basically.
I see.
But in those studies, when they vaccinated sheep against either GDF9 or BMP15, it most definitely shut them down. So it depended on the epitope that was used, but those that were most effective, it did just shut them down. They did not ovulate.
Okay.
So it is so it was able to mimic the mutation, basically. And in fact, the New Zealanders were interested in, developing a vaccine where they might use a very mild adjuvant to mimic the single copy mutation, so that they could actually increase ovulation rate and increase sperm production. And they were mildly successful, but they haven't pursued that anymore. Thank you.
Thanks a lot. Dr. Omapati, if you have any.
Yeah, thank you.
To mention. Yeah. So great. So finally, thanks to all speakers of this session. Despite being in different time zone, they really made it, and they made it in time. So I'm sure it was really hard for you guys, but thanks a lot for being here, and it was really helpful. So I conclude the session. Just two announcements that a participant can write the feedback through the link. There's a link in chat box, and tomorrow we'll meet at 9:30 hours Indian Standard Time. So see you guys. Thanks.
All right. Thank you very much.